Potential, the spark of neural communication. Caitlin Zapola, November 7th, 2025. Focusing on the mechanisms and function, Burke and Bender investigated how the axonal membrane's organization had a direct impact on the neuronal communication. The work they conducted within their research emphasized the membrane is not just a stable boundary, but instead a dynamic and functional landscape, which includes the ion channels specialized to regulate the electrical impulses and action potentials which travel along the axon. The research was focused on the voltage-gated sodium, potassium, and calcium channels, which impact the speed of the neuron's firing and the signal propagation reliability and neuron adaptability. Burke and Bender published their research findings in 2019 in Frontiers in Cellular Neuroscience, a peer-reviewed journal specializing in the neuronal function. Their research directly coordinates with the larger picture of the membrane, contributing to a cell organization by creating the microdomains within the axon, allowing controlled signal timing and efficiency, which directly impacts the brain function and the intracellular coordination. Burke and Bender explained that the axon initial segment, the AIS, is a specialized region of the membrane where the action potentials will originate. Inside of the AIS, sodium channels are packed densely to start depolarization, while potassium channels restore the resting potential and determine the firing frequency of a neuron. Both Burke and Bender emphasize the specific arrangement within the axonal membrane, allowing the neurons to transmit the information rapidly and efficiently. Furthermore, Burke and Bender describe how the calcium channels along the axon is the influence for the release and plasticity of the neurotransmitter. The G protein coupled receptor, GPCR pathways, are the regulation for this action, and these pathways open and close the channels due to the response of the neuromodulators, such as dopamine or serotonin. This mechanism demonstrates how signaling molecules directly interacts with the membrane's environment. Here on Figure 1, directly from Burke and Bender's research, we have 3 different examples. Our first example here is going to be the schematic of the axon compartment. So we have the axon initial segment here, we have the axon, and then we have the terminal. Here in part B of Figure 1, we have the GPCR neuromodulation of the voltage-gated ion channels. That is also from part A. So this is a deeper dive into part A. You have the sodium channels being shown as red, calcium channels being shown as green, and then the potassium channels being shown as blue. Now here it shows as the channels are opening at the end, you have either bars or you have circles. These either show the increase or a reduction due to the neuromodulations, which, as we said earlier, are either the dopamine or the serotonin. The research study conducted by Burke and Bender is important due to the demonstration of the membrane organization determining the processing of information for the neurons. Both authors show when ion channels are localized within the axonal membrane, the neurons are able to stay consistent in their times of spiking and controlling the release of the neurotransmitters. However, when the organization is disrupted within the membrane through either injury or disease, the electrical signal becomes erratic, which can lead to possible neurological dysfunction. Burke and Bender further explained the neuromodulation at the membrane levels, allowing the brain to have the ability to adapt in different environments. Through GPCR control of calcium and sodium channels, the axon is able to adjust the strength and frequency of the signals, furthering allowing learning, attention, and behavior flexibility. The research presented shows a large emphasis that neuronal membrane is a dynamic signaling platform and not just a barrier for protection. The finding from Burke and Bender in 2019 highlights how eukaryotic cells are able to organize their membranes into specialized domains, allowing complex and coordination within the communication, defining that neuronal physiology. Here in Figure 3 from Burke and Bender, in Part A, shows the schematics of the GCPR modulation of the calcium. In Part B, this is going to show the reaction. So on the left side here, we have the reaction that is indicating the calcium that's influencing the release due to the influx. In the center, this shows the two possible outcomes from being reduced. So on the top here, we have where the calcium is coming in and the reduction is then being reduced. However, the bottom center shows the reducing of the influx of each of the channels, where the reduction up here is just from one of the channels. On the right-hand side of Part B of Figure 1, it shows the different calcium regulations of the postsynaptic potential. So the redness here is going to show the calcium potential. However, the black line here is going to be that baseline. This figure shows the organization drives the precision. The ion channel modulation definitely shows the adaptive signaling. And if there is a disruption, that can possibly lead to those neurological disorders. This reference slide credits the scholarly source discussed from Burke and Bender and explained through the presentation, along with the figures used during.